What if everything we thought we knew about life in our solar system is about to change forever? NASA scientists have just unveiled what could be the most earth-shattering discovery in human history. And it's hiding in a chunk of Martian rock that might rewrite our understanding of life itself. But here's where it gets controversial. Even the researchers aren't entirely sure what they've found. In a revelation that has sent shockwaves through the scientific community, NASA's analysis of an ancient mudstone sample collected by the Perseverance rover reveals compelling evidence that microorganisms may have once thrived on the red planet. The specimen, extracted from the Sapphire Canyon core during July 2024, contains mineral signatures and structural patterns that mirror those created by living organisms here on Earth. Yet, and this is the part most people miss, the same evidence could potentially be explained through purely chemical processes that have nothing to do with biology whatsoever. This discovery represents the most promising evidence we have ever encountered in our quest to find life beyond Earth, declared Sean Duffy, NASA's acting administrator. The potential biosignature we've identified on Mars could fundamentally transform our comprehension of the red planet's history and its capacity to harbor life. Duffy emphasized NASA's unwavering dedication to maintaining rigorous scientific standards as the agency works toward its ambitious goal of landing American astronauts on Martian terrain. The sample originated from a remarkable rock formation dubbed Chevaya Falls, situated within the Retva Vallis, an ancient waterway approximately 1,300 feet in width that once channeled flowing water into the vast lake that filled Jezero Crater billions of years ago. Following the drilling operation, Perseverance carefully preserved the specimen in a sealed container, preparing it for a potential future mission that would transport it back to Earth, where sophisticated laboratory equipment could conduct analyses far beyond the rover's current technological capabilities. Decoding the secrets within Martian mudstone, Dr. Joel A. Hurwitz from Stony Brook University, who led this groundbreaking research, describes a fascinating discovery within the fine-grained mudstone, distinctive circular formations the team has informally nicknamed leopard spots, accompanied by small mineral nodules embedded throughout the layered sedimentary material. Using Perseverance's advanced SHERLOC and PIXL analytical instruments, the research team successfully mapped the distribution of organic carbon compounds intertwined with phosphate, iron, and selene, and safer elements arranged in remarkably consistent, repetitive patterns throughout the sample. Two specific minerals have captured the scientists' attention: vivianite and greigite. Vivianite represents an iron phosphate compound, while greigite consists of iron sulfide typically associated with iron and sulfur chemical cycling in environments lacking oxygen, conditions that often support microbial life. These distinctive characteristics appear exclusively in rocks that formed through water-based sedimentation processes, never in volcanic materials. The Martian location sits along the Bright Angel Outcrop System, a series of rock formations that preserve geological layers and mineral veins, indicating gradual chemical transformations that occurred long after the original mud deposits settled into place. The textural evidence and chemical composition point toward low-temperature reactions that reorganized elements already present within the ancient mud. This temperature detail carries crucial significance because moderate thermal conditions create environments where life can potentially survive and flourish, whereas extreme heat typically destroys the delicate chemical signatures that organisms leave behind. Earth's microbes leave remarkably similar fingerprints. Here's where the story becomes truly intriguing. On our own planet, vivianite commonly develops in locations where microscopic organisms reduce iron within water-saturated sediments, subsequently trapping phosphorus within distinctive blue-green nodular formations. extensive laboratory research and field studies have documented biologically mediated vivianite formation through a process called extracellular electron transfer. Essentially, microbes manipulating their chemical environment to extract energy for survival. Greigite frequently manifests in environments where sulfate-reducing bacteria drive complex chemistry within oxygen-depleted mud deposits. In carefully controlled laboratory experiments, researchers detected greigite formation only in living biological systems after several months of incubation, never in sterile, non-biological conditions. The Martian rock sample displays vivianite-rich outer rings surrounding small cores enriched with greigite. This distinctive bullseye pattern perfectly matches the sequence of electron transfer reactions observed in certain Earth sediments where microbial communities actively process nutrients and energy. But here's where it gets controversial. None of this definitively proves that metabolism occurred within the ancient Bright Angel mud deposits. Instead, it demonstrates that the chemical conditions were precisely right to support such biological processes. This subtle yet critical distinction explains why scientists maintain cautious language when discussing these findings. Navigating the complex evidence for Martian life. A potential biosignature represents a geological or chemical feature that might indicate biological origins, but requires additional data to eliminate non-biological explanations. NASA directs mission teams and the public toward the confidence of life detection, cold scale, which promotes systematic, staged claims supported by independent verification. The cold approach follows a straightforward methodology. First, detect an anomalous signal. Then eliminate contamination possibilities, systematically address alternative explanations, and only then discuss Martian life with high scientific confidence. The Bright Angel research occupies an early position on this evaluation ladder. While it successfully clears several necessary verification steps, it leaves the most demanding analytical tests for future Earth-based laboratory investigations. Organic compounds can arrive on planetary surfaces through meteorite impacts or form through purely chemical processes without any biological involvement. The research authors acknowledge these alternative pathways and outline how future analyses could definitively distinguish between biological and non-biological origins. Organic signals without scientific certainty. Perseverance's instruments detected organic carbon compounds in multiple locations throughout the Bright Angel outcrop. The distinctive reaction boundaries rimmed with vivianite surrounding cores richer in greigite create a pairing entirely consistent with iron and sulfur cycling processes documented in the published research. Mineral veins throughout the formation include calcium sulfate deposits, while the mudstone maintains its fine-grained structure with notably low concentrations of magnesium and manganese. Crucially, there are no indicators of intense heating that would have reset the rock's chemistry or scrambled the microscopic textures that preserve potential biological signatures. The outcrop exists within layered sedimentary deposits created by water flowing through the ancient Neretva Vallis channel. The waterway's substantial width, roughly a quarter-mile across, suggests sustained long-term flow into Jezero Crater's prehistoric lake system. While low-temperature conditions favor life-compatible chemistry, they don't necessarily require biological processes. Non-biological organic compounds and mineral reactions can sometimes produce identical shapes and chemical signatures, creating what scientists call false positives in the search for extraterrestrial life. Potential life, not proof of life on Mars. We want to be absolutely clear, this is not life itself, emphasized Nikki Fox, Associate Administrator for NASA's Science Mission Directorate, stressing that the discovery represents a potential biosignature rather than definitive proof of biological activity. Lead researcher Hurlitz echoed this measured approach. We cannot make claims beyond identifying this as a potential biosignature, Hurlitz stated. Other NASA officials similarly emphasized both the discovery's significance and its current limitations. However, this very well could represent the clearest indication of life that we have ever encountered on Mars, added acting NASA Administrator Sean Duffy. This scientific caution isn't excessive hedging for its own sake. It represents how rigorous science avoids false conclusions when addressing questions of such monumental importance to humanity's understanding of life in the universe. Implications for Martian habitability. If the vivianite and greigite minerals indeed formed through microbe-like metabolic processes, then the Bright Angel Formation captures a crucial period when Martian surface waters supported the same chemical strategies that some Earth-based cells employ for energy production today. Such a discovery would extend Mars's potential habitability timeline into a geological window when this region of Jezero crater remained wet and potentially life-supporting. Alternatively, if non-biological pathways created these identical patterns, the rock formation still provides a remarkable record of how iron, sulfur, and phosphorus underwent complex chemical organization within ancient Martian mud deposits. This scenario offers invaluable insights into how Mars cycles essential elements without requiring biological intervention. Either outcome contributes significantly to our broader understanding of planetary evolution. Mars didn't simply undergo gradual desiccation. It experienced fundamental chemical transformations over geological timescales. And these preserved samples allow researchers to track those changes layer by layer through deep time. The research also identifies specific measurements needed for future investigations, isotopic ratios, microscopic textural analysis, and detailed structural characterization of carbon compounds within the core sample can potentially separate genuine metabolic signatures from chemical imposters, advancing the search for life on Mars. The research team has outlined comprehensive laboratory experiments and Earth-based field studies designed to test whether non-biological reactions can reproduce these distinctive textures and mineral combinations under controlled conditions. They've also identified specific analyses that require the physical sample in a pristine Earth laboratory environment, including isotope ratio measurements that biological processes typically influence in characteristic ways. Sample return mission planning will determine how quickly these critical tests can be conducted. Meanwhile, Perseverance continues mapping where these intriguing features cluster and how they relate to surrounding rock formations. The PIXL and SHERLOC instruments have demonstrated sufficient analytical sensitivity to guide this ongoing search effectively. The combination of elemental mapping and Raman spectroscopy detection provides consistent results that can be applied to other outcrop investigations. As scientists catalog new targets, the COLD framework helps communicate research progress without exceeding what the current data actually supports. This methodical approach transforms potential biosignatures into results that the global scientific community can trust and build upon. The complete study has been published in the prestigious journal Nature, making it available for peer review and independent verification by researchers worldwide. And this is where the real controversy begins. Are we looking at the most significant discovery in human history? Or are we once again seeing what we desperately want to see in ambiguous data? The minerals are there. The patterns match Earth's biological signatures perfectly, yet the possibility of non-biological explanations refuses to disappear. What do you think? Are we witnessing humanity's first glimpse of ancient Martian life? Or is this another case of scientific wishful thinking? Should we be more excited about potential life, or more concerned about making premature claims that could damage scientific credibility? The evidence is compelling, But is it compelling enough to change everything we thought we knew about life in our solar system? Share your thoughts. Are you convinced this could be life? Or do you think we need much stronger evidence before making such extraordinary claims?